Good morning, everyone. So today we're going to continue our discussion of the material in Chapter 4. We're going to talk about enzymes. And I wanted to start this off by just reminding you about a little bit about what we were talking about in the last lecture. We were talking about thermodynamics and how principles of thermodynamics govern the reactions that go on in biological systems. And what we're going to talk about today is talking about that, how that connects to the way that reactions are actually occurring in cells. And it turns out that virtually all biochemical reactions that occur in cells have to be catalyzed. They have to be, their rates would be too slow to be meaningful in biology without being sped up by some means. And typically, the means that they're sped up by is by the action of enzymes. And so today what we want to talk about is what enzymes are, what they do, and in particular some of the ways that they can actually enhance the rates of chemical reactions in a way that's meaningful for biology. So this gets back to a question, a very important question that was asked in the last lecture. How is it that reactants get over that energy barrier to products, right? And so that's really what we're focused on today. All right, so I just wanted to start off with a definition. So enzymes are the chemical reaction catalysts of biological systems. And they're usually proteins, but sometimes they're not. Does anybody know what kind of polymer can also catalyze biochemical reactions? I think I heard RNA. That's right. What about DNA? Can DNA ever be a catalyst? I heard no. So it turns out that there's no example that's been found so far of a natural DNA catalyst in a biochemical reaction. But in the laboratory, scientists have been able to make artificial enzymes that are made out of DNA. Okay? So in principle, DNA can also function as a catalyst in certain very sort of specialized kinds of reactions. Okay? But very typically, enzymes are proteins. And one of the reasons that there's been a long history of interest in enzymes is because it turns out that many inherited disorders result from mutations that occur in critical enzymes so that they no longer function correctly or they don't function at all. And then that leads to a disease phenotype that can be inherited because that mistake in the protein sequence is actually encoded in the genetic information. So it can be passed down from parent to child, right? And so that can be an inherited, a way that a disease can be inherited is through that kind of mutation. Okay, and then it turns out that also in medicine, we can use enzyme activities to analyze the state of a person's physiology. We can try to understand what kind of a defect they might have that might be causing disease by measuring the activity or presence of enzymes in their bodily fluids. Okay, so very important in both sort of basic biology, but also in medicine and other areas as well. So fundamentally, what enzymes do is they speed up the rates of chemical reactions. They speed up the rate at which a reactant is converted to a product. And very importantly, they don't change the equilibrium of that reaction. Okay, and we'll talk more about that. Okay, so they are not changed themselves in the process of the catalytic of catalyzing the reaction. Okay, and so very typically enzymes are proteins that have a folded structure that has in the folded three-dimensional structure a binding site for the substrate or substrates for the reaction. And this is what makes enzymes so amazing as sort of chemical entities, is that they are incredibly specific. They don't randomly catalyze reactions in many different substrates typically. In fact, most enzymes are very, very specific. They have a binding site for a particular kind of substrate, and even a very small chemical difference in a molecule that makes it look similar to the enzyme substrate but not identical chemically can mean the difference between the enzyme binding to that molecule versus not binding to it. Okay, and so I've been using this word substrate. So these are typical of the reactants, molecules that can bind in the active site of an enzyme for the chemical reaction to take place. So we can write a very simple kind of equation for an enzyme catalyzed reaction in which we write the enzyme combines with the substrate to form an enzyme substrate complex. And then this is where chemistry actually occurs. The substrate is converted to the product, and then the product is released from the enzyme in the last step. And notice that nothing is happening to the enzyme here chemically, right, overall. So the enzyme molecule that I start out with at the beginning of the reaction is exactly the same chemically as the enzyme that comes out the other end. It's not changed in the course of the reaction. What gets changed is the state of the substrate, right? So chemistry is happening to the substrate, but not to the enzyme. Okay, so enzymes remain unchanged after the reaction. And as I mentioned, so very importantly, enzymes don't change the equilibrium of the reaction. They just change the rates at which the substrate is being converted to product. Okay, so and notice that these are all, in principle, reversible reactions, but of course, if the equilibrium of the reaction lies far to the right towards products, then ultimately we end up with more product than substrate. Okay, and so enzymes can have typically a very profound effect on the rates of reactions in biology. And these are just a few examples. So this is showing you a list of some of the enzymes that are found in various steps of metabolism and other reactions in cells. And what I'm showing you over here is the rate of enhancement that the enzyme is able to achieve for those reactions over the uncatalyzed rate. So in the slowest case, we're going at 10 million times faster than the uncatalyzed rate, right? And it just goes up from there. So you can see from this list, and this is just, a few examples, but many enzymes would fall into this sort of range, that enzymes have a really profound effect. So if you were asked, what's a typical rate at which an enzyme stimulates a reaction above the uncatalyzed rate? Is it 10-fold, 1,000-fold? Those would be too low, right? So typically it would be like 1,000,000-fold or more, okay? And also you might notice that all of these enzymes end in ACE, okay? And that's very typical. So enzyme names typically end in ACE. So if you see that suffix on a name, you can be pretty sure that that is probably an enzyme. Okay, and so back to these. So we looked last time at a reaction coordinate diagram like this, where we had free energy plotted on the y-axis versus the reaction, the course of the reaction plotted on the x-axis here. And remember that free energy is basically the energy that's available to do work, right? It's the energy available to do work. And so if we start off, for any given reaction, we have starting reagents, substrates for the reaction. They start off at some given value of free energy, right? So that's plotted over here. And then they're going to end up over here as products. But to get from substrate to product, very typically there is an energetic barrier for getting over this, sort of getting through this reaction. And this is very typically called the activation barrier. During the course of the reaction, the substrate goes through what's called a transition state to get from substrate to product. Okay, and so and you can see in this example that for the uncatalyzed reaction, which is this black line here, substrate starts off at some value of free energy. It goes through the transition state, and it ends up as product, which has, in this example, a lower free energy than the substrate. So again, is this a spontaneous reaction? Yes, I think I heard yes. So yes, it is. But is it a fast reaction or a slow reaction? Actually, we don't know, right? It could be fast or slow. This actually doesn't tell us anything about the speed, okay? But we can imagine that it might be, certainly the uncatalyzed reaction would be slower than the catalyzed reaction, and that's because you can see in this drawing that the transition state, the energy of this going through this transition state is higher than what you see down here for this little transition state that occurs for the enzyme catalyzed reaction. And so the way they've drawn this in this diagram is to show you that if you think back to that equation where we have enzyme binding to substrate, it forms an enzyme substrate complex. So when that happens, so there's a little bit of an energetic barrier to this, but it's a lot more favorable than this. And once that happens, there's kind of a local free energy minimum that occurs here. So that's a favorable, that tells us that's energetically a favorable interaction. And then something about that interaction actually makes it more energetically favorable. In other words, there's a lower energetic barrier here to get from substrate to product, right? So it's easier now, once the substrate is bound to the enzyme, for it to be converted to product. And again, once that conversion occurs, there's a little local free energy minimum here for the enzyme product complex, and then the product is released, and we end up with this. So the start and finish of this are exactly the same, but what's going on in the middle is different for the uncatalyzed versus the catalyzed reaction. And so, you know, people have been interested for a long time and still are in understanding how is it that enzymes can do this, right? It's quite amazing if you think about it. They're very, very specific, for one thing, much more specific than typical small molecule catalysts that you might use, or just applying heat to a reaction, which can typically speed up the rate of a reaction, but kind of non-specifically, right? So, but enzymes are very specific, and they have this, as we looked at on that chart, a huge, often a very large stimulatory effect on the rate. How can this be possible, right? So people have been very interested in this. And one idea about the way that enzymes do this is that they bind better to the transition state, which is often a chemical state that's somewhere in between the substrate and the product of the reaction, than they do to the substrate. So there's actually an advantage, or an energetic advantage for the substrate to be converted into the molecule that looks more like the transition state when it's bound to the enzyme, because of favorable interactions that occur in the binding site of the enzyme. And we'll talk a little more about how that actually works. But I wanted to point out that there's a very important relationship between free energy and the equilibrium of a reaction, okay? And this is just, I think it's a useful way to think about the relationship between whether a reaction is spontaneous or not, and where the equilibrium lies, and what the free energy change associated with that reaction can tell us about equilibrium. And so that's shown right here. So the equilibrium, we can just write a very simple equation that, describes the equilibrium constant, this KEQ value, for any reaction, in which we just take at equilibrium, if we allowed a reaction to go to completion, so we're at equilibrium, the concentration of product divided by the concentration of substrate. That gives us the equilibrium constant for that reaction. And for any reaction, this number is going to be different, right? For some reactions where the reaction equilibrium lies far to the right, in other words, we get at equilibrium, we have a lot more product than substrate, this is going to be a big number, higher than 1, of course, big, but might be a very high number. And if at equilibrium, the equilibrium lies the other way, where we favor substrate over product, then in this ratio, of course, that's going to be a value that's less than 1 at equilibrium. Okay, and so then there's a very simple relationship between the free energy of the reaction and the equilibrium constant here, which is given in this equation. All right? And what this tells us is that if the equilibrium constant is 1, in other words, we have at equilibrium we have the same concentration of substrate and product, then the free energy change associated with that reaction is 0. if the equilibrium constant is higher than 1, then the free energy that is associated with that reaction is negative. Okay, that means that, again, and you remember from last time that a negative free energy change means that the reaction is spontaneous, right? It is that products are favored over reactants over time. If, on the other hand, the equilibrium constant is less than 1, like here, then the free energy change associated with that reaction is positive, and that's a reaction that is not spontaneous, right? At equilibrium, we favor substrates over products. And again, you can think about it in terms, I like to think about it in terms of these free energy diagrams because it's very visual, it's very easy to think about it. If you think about the free energy associated with the substrates and products, right, where are we ending up? Are the substrates ending up at a higher or lower free energy than the substrate? Okay, and that'll affect the equilibrium of the reaction. Again, it doesn't tell us anything about the rate of the reaction. If I tell you that a reaction is spontaneous, and I ask you, and how fast is that reaction, can you say anything about the rate? No, right? You can't, right? So it could be fast or it might be slow. You don't know. But what all you know is that if I tell you it's spontaneous, you know that the free energy change associated with that reaction is negative. So over time, given enough time, if it's a slow reaction, products will be favored over substrates. Okay, so as we said, so every chemical reaction that involves chemical bonds being broken and reformed involves an activation barrier of some sort, right? And so we can call this the free energy of activation. Sometimes it's abbreviated like this. And if we have a reaction that is occurring in an uncatalyzed way, we have no catalyst or enzyme around to speed up the rate of the reaction, then very typically the energy required to get from substrate to product, to get over that activation barrier, just comes from thermal motion, right? It comes from heat. So every now and then there's enough thermal motion that a bond that needs to break in the reaction can break, and then a new bond can form. So you can imagine that if we're going to wait around for enough thermal motion to break bonds that are occurring in a biochemical reaction, we might have to wait a really long time for one thing, so it might not be so relevant in the lifespan of an organism. And furthermore, if heat were used to catalyze or speed up the rates of reactions in biology, it's not specific, right? So we're speeding up sort of lots of things, not just a specific reaction. And furthermore, if we heat things up high enough, then we start to disrupt cellular structures and denature enzymes and things like that, right? So, or denature whatever, other proteins and other biomolecules. So that's not an effective way typically for reactions to be speeded up in biology. That's why we need enzymes. Okay, so back to this. So here's this is this free energy versus the course of the reaction kind of plot. And again, we start off with reactants or substrates here, and note that these reactants it doesn't have to be I showed you this very simple example of a single substrate molecule being converted into a different product molecule, but actually in many cases in biological reactions, we're looking at 2 or sometimes even more than that molecules coming together and reacting. And so in this example here, we might have 2 different molecules that are binding to an enzyme active site. They have to go get over this free energy of activation, this barrier through the transition state. This is trying to indicate that in the transition state, bonds are breaking and new bonds are forming, and then those products are ultimately released. And in this example, the free energy change of this reaction is less than 0. The products end up at lower free energy than the reactants, and so this would again be a spontaneous reaction, right? This is a favorable reaction, and we end up with new molecules at the end. Okay, and so enzymes lower that activation barrier. They don't change the free energy associated with the reaction, right? They increase the rate. They don't change that free energy, that overall change in free energy that occurs. So they're not changing this value or this value. What they are changing is this value, right? Usually lowering this value of the energy of activation to some lower value, so it makes it easier to get over this hump. Right, and so this is shown right here. So if we look at a comparison of the exact same reaction, in one case it's catalyzed by an enzyme. In another case, it's uncatalyzed. And this is sort of what this is showing you. The black line is the course of the reaction in the absence of the enzyme, right? So here's the energy of activation, and that's what this arrow is indicating. This is the energetic barrier that reactants have to get over to get to products. And if we have an enzyme present, that barrier is less. And so we lower this barrier. It's easier to get from reactants to products, but we haven't changed the starting energy of the substrate or the ending final energy, free energy associated with the products, right? So the overall delta G of the reaction is unaffected by the enzyme. So be sure that you really think about this and study it and look at these diagrams, because every year a number of students get confused about this, and they start to think that enzymes are changing the overall equilibrium of a reaction or the free energy of a reaction, and really they're not, right? They're just affecting the speed at which we get from from the starting material to the ending material, okay? Okay, so again, we talked about what substrates are, and we talked about the formation of an enzyme substrate complex. And another term that I want you to be familiar with is the term active site. So this is used a lot to describe enzyme binding sites for their substrates and where chemistry actually occurs, right? So this is where the substrate binds and where chemistry happens. It's the active site. It's the part of the enzyme where it's the real business part of the enzyme. That's not to say the rest of the structure of the enzyme is not important, but the real chemistry is going on in the active site. And so that's where we often want to focus our attention if we're trying to understand how a particular enzyme is speeding up the rate of a reaction. Okay, and we can also talk about this concept of induced fit. And so what this means is that very typically, and we know this from all sorts of experiments that have been done using crystallography, for example, where we can look at the structures of enzymes before they bind a substrate, after they've bound a substrate, and then after they've converted the substrate to product, and compare those different structures. And what we often see in those kinds of studies is that enzymes start off with some three-dimensional structure. They bind to a substrate, and when they do that, there's kind of a repositioning of chemical groups in the active site that then puts them in a position to enhance the rate of a reaction or help orient the substrate so it is aligned for the chemical reaction that needs to occur. Okay, so in other words, that means that enzymes we know now are not rigid molecules that always look the same in the course of a reaction. They can actually change their structure a little bit, sometimes a lot, but usually in sort of subtle but important ways, so that they can bind a substrate, chemistry occurs, and then release the product. And this is an example. This is actually from a figure out of the textbook, your textbook, that shows the structure of an enzyme called hexokinase. So this was one of the first very early crystal structures that was solved in Tom Seitz's lab at Yale. And so what Tom and his colleagues were able to do was to crystallize this enzyme, which is a key enzyme in metabolism. And what they found out was really interesting, because they figured out that this is an enzyme that has, an overall three-dimensional structure associated with its activity. It's got a very obvious sort of cleft here that turns out to be the active site. So this is where substrates can bind. And when the substrate binds, there's a change in the conformation. So if you compare this unbound structure of the enzyme to what happens when binding occurs, you can see that actually the substrate induces this change in the cleft. So there's actually a closure around the substrate. and now it's in this little pocket, and that's when chemistry can happen efficiently. And as I said, that's not unique to hexokinase. Many enzymes operate this way. So this shows an example of a reaction. We have a sort of a catalytic cycle of enzymes. In this case, we're talking about another enzyme, in this case, an enzyme called beta-galactosidase. And this is an enzyme whose job is to cleave a disaccharide, right? So it's a two-sugar molecule. It breaks this glycosidic bond. So it has a binding site for this disaccharide, and it's very specific, right? It's this particular disaccharide, not others, can bind here. And remember that when we break a glycosidic bond, that's a hydrolytic reaction, right? It's a hydrolysis reaction. We're adding water across that bond. Water comes in, chemistry occurs, that bond breaks, and then the enzyme releases the two individual sugars, galactose and glucose, get released. But the enzyme itself is not changed in the course of the reaction. It remains functional. It now has empty active sites, and it's poised to bind to another disaccharide and catalyze this reaction again. So an enzyme can typically catalyze many, many reactions per enzyme molecule, and we call that turnover, right? Turnover. So for a given enzyme molecule, we can have many, many substrate molecules that get reacted per enzyme molecule. And so there's several different ways that binding of a substrate and the energy associated with that favorable interaction in forming the enzyme-substrate complex can lead to catalysis. Okay, so, and I want to just point out what these are. So first of all, there can be a reduction in entropy. And what that means is that, and you might be thinking, wait, but entropy, increase in entropy is favorable. Yes, that's true. But on the other hand, in a chemical reaction, very typically, and you probably remember this from chemistry class, the chemical bonds have to be oriented correctly for chemistry to occur, right? And so one of the ways that that happens is by alignment of substrates in the enzyme active site such that they're positioned correctly for chemistry to occur. And another way that entropy is often reduced by an enzyme is by stripping off water molecules that might be associated with an enzyme, or reorganizing water, right? And so temporarily, the entropy can be reduced in that process by orienting or changing the structure of the molecule and the waters that might be associated with it. And then that leads us to the second way that binding energy contributes to catalysis, which is substrate desolvation. So again, remember that these are reactions that are occurring in an aqueous environment. There's lots of water around. These molecules, substrate molecules, might have lots of hydrogen-bonded waters that would otherwise prevent them from efficiently reacting and being converted to product, either reacting with another substrate or just being converted to a product if it's a not a bimolecular reaction. And so desolvation is another way that the enzyme interaction with the substrate can lead to a more efficient reaction than in the uncatalyzed state. And finally, this idea of induced fit, where these, you can see these are sort of all, there's sort of relationships between these, but the induced fit idea is that, again, you have a substrate that might have lots of degrees of freedom in solution, right? It has lots of, there's maybe lots of dynamics of that molecule. When it binds the substrate, it's now constrained. So not only does binding constrain the enzyme, but the enzyme constrains the substrate as well, right? So there's that sort of, again, the idea of induced fit with the interaction between an enzyme and a substrate. And so once binding occurs, so those are all some favorable things that can happen upon binding, then chemistry happens. And the way that this occurs and is favored by the enzyme, again, there's sort of a number of principles that have been learned over the years from studying many different kinds of enzymes and how they function. And there's sort of 4 different ways that generally are employed by enzymes to enhance the rate of a reaction, right? So one is orienting substrates correctly. So we already kind of talked about that. Straining substrate bonds. Now this is the idea that in lowering the reaction barrier, and this is a concept that's been sort of looked at over and over in different contexts over the years, it turns out that enzymes often actually bind more favorably to the structure of the transition state in the reaction than to the substrate or the product, the ground state of the substrate or the product. And so that means that when a substrate binds to an enzyme that's operating this way, there's actually some strain that happens in the substrate molecule. And often that strain is associated with moving the molecule energetically closer to the state where a bond can actually break or a new bond can form. Okay? So that's another way that enzyme active sites operate, is by straining substrate bonds. And in doing so, they provide a favorable microenvironment. So again, this means that this is associated with this idea of stripping off waters, straining bonds, orienting substrates correctly so that they're in line for a chemical reaction to occur. All of those things will enhance the rate of the reaction. And occasionally, so these are all sort of things that happen typically with non-covalent kinds of interactions, occasionally there's covalent bonding to the substrate. And this is, if this happens, this is a transient thing. So there might be transiently a covalent bond formed between an active site amino acid in the enzyme and the substrate. Chemistry then occurs, and when the chemistry occurs, that transient covalent bond breaks and the enzyme is once again restored to its starting state. Okay, so when there's covalent catalysis, it doesn't mean that the enzyme is changed forever. It just means that it's temporarily changed during the course of the reaction. Okay, and so it turns out that in looking at many different active sites over the years and different kinds of enzymes, there are specific functional groups that are associated with active sites, and some of them are shown right here. Okay, and so we can have functional groups that are acids or bases, and that leads to what's called general acid-base catalysis in enzyme active sites. We can have residues like cysteines or sometimes histidines that can lead to, or even serines, that can lead to covalent catalysis. So this is the idea that we just mentioned, where you can have a transient formation of a covalent bond in an active site that leads to, that can stimulate the rate of a reaction by, again, making it easier for the substrate to be converted to product. And sometimes there also are metal ions that will bind to some of these kinds of residues in active sites, and then those metal ions can help speed up the rate of reaction because, again, they're oriented in a very specific way with regard to the positioning of the substrate. All right, and so in addition to amino acid side chains that are found in the enzyme, it turns out that many enzymes also use what are called cofactors. So these are non-protein groups that bind to the enzyme and help stimulate the rate of a reaction. They can be metal ions, often things like magnesium or copper or iron. So think about it. These are often metals that we need in our diet in trace amounts, and that's because we have enzymes that require these ions for catalysis. Okay, that's typically what they're doing. In other cases, there are what are called coenzymes. Coenzymes are not metal ions, but they're small molecules, small organic molecules. They're typically vitamins, right? So when you take your vitamin pill, what you're doing is supplying cofactors, or the building materials for cofactors, to your body that are used in enzymes. And these often, they can bind loosely to enzymes, or they can be very tightly bound. If they're very tightly bound, then we typically will call them prosthetic groups, right? It's like having an artificial arm prosthesis. It's really an integral part of that enzyme if it's a prosthetic group, and it really probably never comes off effectively, okay? Okay, and so then there's a number of factors that can affect enzyme activity and can control the rate at which an enzyme catalyzes a reaction. And we want to just talk a little bit about some of those factors because they turn out to be very important in figuring out how fast a reaction is going inside of a cell or an organism. So first of all, the concentration of an enzyme if we were to plot the rate of a reaction as a function of enzyme concentration, and we have a given amount of substrate, and in this example we have a very high amount of substrate. So this is, let's say we start off with a really high concentration of substrate with respect to the enzyme. As we increase the enzyme concentration, we have more and more enzyme molecules available to catalyze the reaction with the substrate. And so we see that there's a linear relationship, right? And if we, eventually if we got up high enough, this is going to level off, because if we have, eventually we have so much enzyme around that there aren't enough substrate molecules to interact with those enzymes, and then this relationship will level off. Okay, but initially, if we have a huge excess of substrate over enzyme, this is a linear relationship. And the substrate concentration also affects the reaction rate. So if we have a constant amount of enzyme in a reaction, and we start titrating in substrate to that reaction, initially we have these very low concentrations of substrate. We have very few substrate molecules for the amount of enzyme that we have. So most of the enzymes aren't finding a substrate to react on, right? But some of them are. And as we increase the concentration of substrate under those conditions, we just see faster and faster rate of conversion, because more and more of those substrate, enzyme molecules are now able to find a substrate to bind to and catalyze the reaction with, right? So that is this relationship right here. But eventually, we get up to a high enough concentration of substrate that we've saturated all of the enzyme molecules that are present. So now, essentially, every enzyme molecule in that reaction has a substrate bound to it, and once it catalyzes the reaction and releases the product, there's another substrate waiting right there to be bound, right? And so we're going as fast as we can under those conditions. And so that's what's called saturation, and that's what we see occurring in this example right here. Okay, so both substrate and enzyme concentration can affect reaction rates. And then, of course, there are all sorts of ways to inhibit enzymes as well. And you can imagine many scenarios in biology where this might be advantageous to inhibit an enzyme, right? So this happens all the time in metabolic pathways, for example. You want to slow down the rate of an enzyme because you don't want to make too much of a particular product, or you want to, there also are activators, right? So you want to activate an enzyme. You can have the sort of the reverse going on. And your book talks about two specific ways that inhibition can work on an enzyme. Competitive inhibition, and this, I think this is sort of conceptually a very straightforward idea, right? You have a competitor, you have a molecule that looks a lot like the substrate, and it looks enough like it that it can actually bind in the same active site where the substrate can bind. And when that happens, it just literally competes for binding. And so if there's lots of inhibitor around that works this way by competitive inhibition, you have the active site occupied by the competitor, substrate can't get in there, and therefore the enzyme is effectively inhibited. We don't see as much conversion to product in the presence of that kind of inhibitor. And then another way that inhibition can occur is by what's called non-competitive inhibition. And what this means is that we have an inhibitor that, rather than competing directly for binding in the substrate active site, it actually binds somewhere else on the enzyme, right? It's found some other cleft or something to bind to, and by doing so, it induces some other kind of change, maybe in the structure of the enzyme, that leads to an inability of the enzyme now to bind effectively to substrate. And so then we don't get as efficient a reaction under those conditions with a non-competitive inhibitor. But they work differently, right? So competitive and non-competitive inhibition are two very distinct kinds of mechanisms. And this is just a cartoon that illustrates this. So again, with competitive inhibition, we have an active site that can bind to a particular kind of substrate, and when the competitive inhibitor is present, it's able to occupy the same site. And so that means that now the substrate cannot bind, and so the reaction is effectively blocked under those conditions. And then in contrast, if we have a non-competitive inhibitor, the idea is that we have a binding site that's not the same as the binding site for the substrate. It's somewhere else on the enzyme, but nonetheless, the effect is to change the ability of the enzyme to interact with the substrate because maybe there's been a structural change in the binding pocket that normally would recognize the substrate, and it can't bind as well when that non-competitive inhibitor is present. Okay, and so lots of drugs, it turns out, so lots of small molecule drugs that are used actually work this way. This is how they work. They either are competitive inhibitors of a specific enzyme, so they look a lot like the substrate for a particular enzyme, or they are non-competitive inhibitors. They bind somewhere else on the enzyme, but nonetheless, it can be very selective, right? And they can block a particular enzyme's activity. And so cells are constantly regulating the activity of various kinds of enzymes. And that's because in metabolism, and you think about sort of what's going on in a cell, you don't want these reactions going on willy-nilly. They actually have to be controlled in various ways. And that's because depending on the conditions that are present in the cell at a given time or in a tissue or even a whole organism, the organism might need more or less energy. It might need more or less starting materials for making proteins and all sorts of other kinds of, more or less hormones, that sort of thing. And so all of those enzymes involved in those pathways have to be controlled. They have to be regulated specifically. Right? And so we have non-competitive and competitive inhibition going on. We also have various forms of allosteric regulation. I'm going to talk about that now. Going on, and occasionally there's also covalent modification of the enzyme by the addition or removal of different chemical groups, and that's very typically associated with phosphate groups, but it can be other kinds of functional groups as well, like methyl groups, for example. Okay, so allosteric regulation. So the idea here is that there is binding to an enzyme that occurs outside of the active site. So it kind of sounds like non-competitive inhibition, right? Binding is not occurring directly in the active site. It's somewhere else. But very typically, this is associated with enzymes that have multiple subunits associated with them. So multiple polypeptides that might come together and form a structure that then is very amenable to allosteric regulation because we can have one subunit binding to a regulatory molecule, and then that influences the effect and the ability of the other polypeptides in that complex to bind to something or to carry out some kind of a reaction. And so when we have enzymes that are controlled by this kind of regulation, allosteric regulation, they very typically have two very distinct conformations. They have a high affinity form in which the enzyme binds very tightly to substrate, strongly to substrate, and then we have a low affinity form in which the enzyme binds to the substrate only very weakly. Question? Is there no middle ground for that? Typically, there's not much middle ground for that in allosteric regulation. It's either or. Okay? And a great example of this that you're going to learn about later is hemoglobin. Okay? Hemoglobin, it carries oxygen in our blood. That is a very wonderful example of allosteric regulation where it either binds oxygen with high affinity or low affinity, and there's a very sharp shift in those affinities. Yeah? Transient coordination. Let's see. Was that to binding to substrate? Sorry. I think what I meant if which right here. Right. So basically, what transient coordination, what that means is that, like this is the case often for cofactors. They bind, but they may not bind very tightly. Sometimes they do. Metal ions actually often bind very, very tightly to enzymes. But sometimes there are other kinds of cofactors that can bind transiently, and that means exactly what you might think it means. It means that those can come onto an enzyme and then they can leave. Okay, so they're not covalently bound, and they might not even be that tightly bound, but they're bound transiently. They can come on and off. Okay. Okay, so here's a couple of cartoons that show allosteric regulation. So when we have an allosteric activator, this means that we have, and I'm not very fond of these drawings from your book, I have to say, because again, it doesn't sort of show you this idea of multiple polypeptides or multiple subunits, and that's really, I think, more of the classic way to think about allosteric regulation. But the fundamental idea is that if we have an allosteric regulator, it binds somewhere else on this enzyme or enzyme complex, and that binding event influences the ability of a distant or different site that interacts with a substrate to bind to substrate. Okay?
Sorry, the multiple polypeptides, are those like the allosteric activators?
No. So what I'm trying to just describe is that typically, and you'll see this when we study hemoglobin, you have a situation where an enzyme is really formed of several different polypeptides that come together. And it's really the binding of one of those polypeptides to a small molecule, like an activator or an inhibitor, that then influences the rest of the subunits, the rest of the polypeptides in that complex to do something, like bind to a substrate. And that's classically what happens with hemoglobin. Right? So what we'll see there is that hemoglobin has multiple polypeptides. It has 4 different polypeptides in that complex, and it's binding of activators or inhibitors to the interfaces of those polypeptides in the complex that affect the ability of the rest of them to interact with oxygen. Okay? And so, but fundamentally what I want you to come away with here is that allosteric activation or inhibition, which is shown on the next slide, is just the idea that we have binding to a site that's not the same as the substrate binding site, but there's an influence on the ability then of the enzyme to interact with a substrate in that case. Okay, so it's very analogous to non-competitive regulation in this case. Yeah?
Do all enzymes have an allosteric site?
No. So do all enzymes have an allosteric site? No, they don't. Right? So some do and some don't. I mean, in principle, you could imagine that you might be able to find an allosteric regulator of many different enzymes, but in practice, many of them don't. Some do and some don't. Okay, and then I want to just introduce a couple of other important concepts. So what we're going to see in metabolic pathways is that very frequently these are regulated by what's called feedback inhibition. And that means that if we have a substrate that's being converted through several steps to a product, that product in many cases can then function as an inhibitor of the first enzyme in that pathway. And very often that turns out to be through an allosteric kind of mechanism, right? So it's not a direct competitive inhibitor of that starting enzyme, but rather it binds somewhere else on that enzyme and affects its ability to catalyze the reaction. Very useful because if an organism has plenty of this amino acid, it wants to slow down or even shut down this pathway so that we don't build up an excess of this over time. Okay? Okay, and finally, we just want to mention that there's chemical modification that occurs of enzymes that involves phosphorylation or dephosphorylation. That means adding a phosphate group to an enzyme or removing a phosphate group from an enzyme. And that can change the enzyme structure and affect its ability to interact with substrates. I see a hand back there. Yes. How do I know if it's non-competitive or allosteric? I mean, there's some overlap in the way those terms are used, actually, right? But typically, allosteric. This is why I'm saying that for the way that I typically think about allosteric regulation, it has to do more with enzymes that are large and have multiple subunits, where you have binding of one subunit or interface between subunits that affects the rest. Whereas non-competitive regulation is, I think of more as associated with a single polypeptide. Okay, your book doesn't make that distinction, but I think that's... Yes, that's probably a reasonable way to state it, yeah. Okay, and then I just want to also point out that temperature and things like pH can affect enzyme activity, and your book has a couple of figures that illustrate this. So enzymes that function in a particular environment have evolved to have an optimal temperature and pH activity at these temperatures and pHs that are associated with where they function. So for a human enzyme, typically our enzymes are evolved to operate optimally at our body temperature. But if we look at organisms that grow at, say, a very high temperature, many of those enzymes have evolved to operate best at those much higher temperatures. Same for pH. If we have an enzyme that works in the stomach where the pH is very low, its optimal pH is much lower than what we see for enzymes that work in other tissues where the pH is higher. Okay, and then your book has, very sadly for me, since this is sort of my field, just 2 very short paragraphs about ribozymes. But you should look at that because it turns out that there are a lot of interesting examples where it's not protein, but it's actually RNA that's catalyzing a biochemical reaction. And perhaps most interestingly, all proteins in our cells and all cells in biology that we know of, for the most part, at least big proteins, are made by the ribosome, which is fundamentally a ribozyme. Okay, so RNA catalyzes the synthesis of proteins, very interestingly, and that has important implications for the way that cells have evolved and the way that metabolism evolved. Some people think that all of the original enzymes in biology were made of RNA. Okay, so have a look at that in your book, and have a good weekend. I'll see you guys on Monday. No office hours today, right? No office hours today. Monday will be office hours as usual.